So, welcome back, everyone. We're very excited to start this next session. You know, the focus of today is the future of MG. And, well, we thought it was very important for this meeting to have a special stage for early and mid-career researchers. So we dedicated this session to a lot of exciting abstracts, and we thought it would be good to inform you also on how the selection of abstracts went. So basically we have this lovely steering committee whom you can see in the booklet, and they helped with assessing all the abstracts. There were, I think, over 240, and each abstract was assessed by at least 2 independent reviewers, and those that were flagged to be very interesting and to present on stage were invited to actually present here. Of course, as always, there's much more talent and interesting things than time, so we had to be selective here and there, but we're very glad to have this this session. So another sort of household remark, we would really want to encourage all the early and mid-careers, doctors and scientists, people here, to ask questions. You're very welcome. Don't feel scared. For all the people here, they usually are very friendly, the neuromuscular crowd, so feel free to ask questions. And this is going to be a rapid session, so all the speakers will follow one after another to give as many a stage as possible. So, am I forgetting anything?
No, we only have about 2 minutes questions, so if you have a question they cannot answer, you please take it to the break.
Yes. So, without further ado, it's my pleasure to introduce Gregorio Spagni from Florence, Italy. He's a neurologist there, and he will tell us about B-cell depletion for acetylcholine receptor myasthenia.
Thank you for your kind introduction, and good morning, everyone. I'm very happy to be here today to talk about the main findings of this study, on which I've worked during my postdoc at the Charité University in the group of Professor Prus, and to the completion of which I'm now working from my current institution, the University of Florence. So, as Professor Payne has shown, I think it's not working, yeah. Chimeric Autoantibody Receptor T cells has emerged in recent years as a novel tool to selectively target antigen-specific B cells driving antibody-mediated disease. And this has been comprehensively shown at the preclinical level for pemphigus vulgaris, MuSK-MG, and anti-NMDA receptor autoimmune encephalitis. Of course, such an approach could also be useful for AChR myasthenia gravis, where, however, the antigenic target and the autoimmune response is more complex. As compelling data shows that anti-alpha-1 and anti-beta-1 AChR subunits antibodies are the main pathogenic drivers, in this project we aim to develop double AChR T cells targeting B cells specific for the alpha-1 and beta-1 AChR subunits, and to evaluate their functionality in vitro and in vivo. So we first designed these 2 CAR constructs, one with the first extracellular domain of the alpha-1 subunit, the other one with the first beta-1 extracellular domain fused with the CD8 transmembrane domain, and for 1BB and CD3Z co-stimulatory and activation domain. These were cloned into lentiviral vectors that were used to transduce healthy donor-derived T cells that were expanded, enriched, and used for the experiments. We tested these conditions, so T cells transduced to express the alpha-1 CAR, the beta-1 CAR, and T cells co-transduced with both vectors, as well as desmoglein 3 CAR T cells or untransduced T cells as control. And during the presentation, this color coding is used for these conditions. So first of all, we could demonstrate CAR T cells, CAR expression on the T cells and binding by human monoclonal antibodies specific for either the alpha-1 or the beta-1 subunits, as shown in these plots. We could demonstrate binding for all these antibodies, 5 anti-alpha-1 and 5 beta-1 reactive monoclonals that were provided thanks to our collaboration with the groups of Professor O'Connor and Professor Derfus that generated these maps. And then we used the variable heavy and light chain sequence to clone them into our surface IgG lentiviral construct and transduce cell lines such as NALM6 and KFX2 to let them express this IgG on their surface, mimicking a B-cell receptor. So that after validating surface IgG expression, we could use these cells as model autoreactive B cells to test CAR T cells functionality. So first of all, we could see that upon co-culture with the target cells, the CAR T switched to an activated phenotype as assessed by measuring the proportion of cells expressing the early activation marker CD69. They secreted effector molecules such as interferon gamma and granzyme B as assessed by ELISA, and they underwent proliferation over 5 days of co-culture in this fluorescent dye dilution experiment, with each peak representing one cell division. And as you can see, all these effects were specific, meaning that the alpha-1 CAR T cells reacted with anti-alpha-1 target, the beta-1 with the anti-beta-1, and the co-transduced with both, with no effect seen in the control conditions. And importantly, over 15 days of ex vivo expansion, we didn't see any substantial change in the expression level of the surface CAR. Then we moved to assess whether the CAR T were also able to deplete their target in vitro in a firefly luciferase assay, where the target cells express firefly luciferase, so the amount of living target cells can be measured by measuring bioluminescence. We tested different effector to target ratios from 1:2 till 8:1 to build then these dose response curves, and that shows that the alpha-1 CAR T cells and the beta-1 CAR T cells were very efficient in depleting their target, also already at the 4:1 E/T ratio, but also that the double transduced CAR T cells could deplete their an oligoclonal mix of anti-alpha-1 and anti-beta-1 target cells with no effects in the control conditions. As we know that CAR T cells are meant to work in an environment where soluble antibodies are present, we then assess whether anti-AChR antibodies could affect their functions. And of course, sorry, I'm forgetting this slide, we could also demonstrate efficient killing for all the anti-alpha-1 and anti-beta-1 target cells. So I was mentioning we wanted to check whether soluble antibodies could affect the CAR T cell functions, and first of all, we could see that the presence of a mix of anti-alpha-1 and beta-1 AChR antibodies, either at 5 or 25 microgram per ml, did not affect the capacity of these cells to deplete their target, as the cytolysis rate was comparable to the no monoclonal antibodies conditions. And also mirroring these results, we could see that the presence of soluble antibodies did not induce substantial interferon gamma secretion, nor induce in vitro proliferation in a shorter 2 days experiment. So knowing this, we then moved to the in vivo model, and in this case, we just completed a pilot experiment of a xenograph passive transfer model, where an immunodeficient NOG mice receive NALM6 cells followed by CAR T cells. In this case, we tested NALM6 cells expressing an anti-alpha-1 surface IgG followed by anti-alpha-1 CAR T cells. And we could see here also efficiency in target cell depletion in vivo. In this model, as you can see, the two curves are separated. We also noticed after day 13 that there is an escape in the AChR alpha-1 condition, but in this case, this was expected, as only 85% of the target cells used in this experiment were actually expressing surface IgG and therefore were targetable. Now the full experiment with both alpha-1 and beta-1 target is now running. Overall, this data shows that AChR CAR T cells can efficiently and selectively deplete target cells in vitro. They are effective against monoclonal but also oligoclonal target cell population, and they're not affected by soluble anti-AChR antibodies in vitro. Preliminary data also support the functionality of alpha-1 CAR T cell in vivo, and the full experiment is now running to assess also the beta and the co-transduce. And importantly, we also showed that the co-transduction of T cells can allow targeting of B cells with different antigenic specificity, which may be a viable option to tackle more complex autoimmune disease such as AChR myasthenia gravis. So we hope that upon further preclinical validation, AChR CAR T cells can pave the way towards antigen-specific therapy in AChR myasthenia gravis. So with this, I've concluded. I want to thank you all for your attention, and of course the CAR T team at the Charité and all our very important collaborators. Thank you.
Thanks very much. The floor is open for questions.
Thank you, Gregorio. Outstanding work. Do you have evidence of B-cell depletion in the thymus or in bone marrow? Are you going to see that?
In this model?
Yeah.
No, we usually check the spleen of the mice to see if there is efficient B-cell depletion there. But yes, it's worth looking also in these other sites for sure, yes.
Okay.
Okay. No more questions? Okay, thank you very much.
Thank you.
Gregorio. We'll call our next speaker. Dr. Michelangelo Maestri Tassoni from Italy, who will talk to us about thymectomy in juvenile myasthenia gravis.
Good morning, everybody. Thank you for the opportunity to share our data, our clinical data about thymectomy in juvenile myasthenia gravis. We all know, also from yesterday's debate, that thymectomy is a well-established therapy for myasthenia gravis with AChR antibodies aged between 18 and 65. But the efficacy of thymectomy in the treatment of juvenile myasthenia gravis is still debated since there is no trial about this age. And some data suggest that it is generally safe and effective also in this population. So what we wanted to do is to analyze retrospectively our data about the long-term neurological outcomes and the safety of thymectomy in our population of juvenile myasthenia gravis patients. So we retrospectively evaluated the clinical, therapeutic, and surgical data of our population that underwent thymectomy in our center from 2000 to 2022. And since it was very difficult for us to find a control group of juvenile myasthenia gravis that can be matched for severity of the disease, we used a kind of gold standard, as I can say, a control group of early onset adult myasthenia gravis patient with a similar sex and disease severity at baseline comparison. And we use MG-ADL, MG composite score, prednisone dosage, and also PIS as the primary outcome. So here you can see our population. We have 29 patients with a female predominance. These are the other clinical data. The age of onset was at mean 12 years. And then you can see that prednisone was quite high in this population. 37.5 was the median dosage. These are the other clinical data. The ADL at the baseline as a median value was 6, but there was a huge differences between patients. Nowadays we use more or less usually the robotic assisted thymectomy, but we have some data also with sternotomy. As you can see, 2 patients had thymoma. We don't have much postoperative complication, and we have a quite long follow-up, and this was very nice in our opinion. So this is the clinical outcome at last follow-up, and in our population, almost 80% has a positive outcome, as you can see. Nearly 20% has a complete stable remission, 16% a pharmacological remission, and almost half of the population minimal manifestation. And then you can see also that as a population, the drug usage is less than at baseline. When we compare the two populations, early onset myasthenia gravis and juvenile myasthenia gravis, the trends are very similar. As you can see, both for myasthenia gravis activity of daily living on the left side and for myasthenia gravis composite score with the first clinical response also in the first year of the disease. And a similar trend, we observe a similar trend also for prednisone dosage. We try to analyze if there were something that could predict the clinical response, and we didn't find much really. We did not find not the myasthenia gravis severity, nor the titer, nor the histology, both as thymoma versus thymic hyperplasia, but also follicle numbers and follicular hyperplasia. This does not differ from the two groups. The only variable seems to be age at surgery. Operating at an age of over 12 years increase the likelihood of myasthenia gravis remission. The other point we want to underline is the safety. We did not observe an increased incidence of autoimmune disease or cancer after thymectomy, and you all know the importance of this point. And in our cohort, the 2 patients with thymoma did not present a recurrence of thymoma in our observation. So looking at the literature is very heterogeneous. In this review, they evaluated 17 studies, globally considering the 588 patients, but with, I would say, an important percentage of clinical stable remission. And in a few studies, I found 4 studies also in this other review, in which they compare surgical and medical treatment for juvenile myasthenia gravis. Surgical treatment has a better outcome without important clinical complication. What we can try to explain why 12 years of old can be a turning point of this significance is probably because it's possible that some activity, some immunological T-cell malmaturation that usually occur in thymus could occur also in extrathymic areas if the thymectomy has been performed even too early. So, coming to the conclusion, we want to underline that probably the effect of thymectomy is similar to early onset myasthenia gravis, also before 8 years of age, but it should be suggested to wait until puberty or 12th year. That not many data are present with mini-invasive robotic approach, but of course now we have to look for mini-invasive surgical approach. And in our experience, there are no problems as concerns neoplastic and autoimmune disease, also in maybe not so long, but quite long follow-up. And I would like to thank, of course, all the neurology team and surgical team in Pisa, and thank you for your attention.
Okay, thank you very much. Are there any questions from the audience? There's one question, a couple of, I see people there. Go ahead.
Thanks for the great talk. I had 2 questions. One, how many prepubertal children did you have in your cohort? Because your average was 11 years and it was standard deviation of 3. And second, how many patients had thymectomy within the first year of onset and how many was after that?
Okay. I have to think about it, but we have, I think, something like 6 or 7 patients that underwent thymectomy before 12 years of age. And usually the mean time from the diagnosis to thymectomy was 1.5 years. So probably we have to check at this point exactly, but we have both early and late, early surgical approach and not so early. But we didn't see any different about this. We tried to put also this variable in the analysis, and it doesn't change so much.
But then you have, your predominant cohort is about 12, and is that the reason of the bias that you're seeing improvement in that? Because to say that, you need to wait till puberty to do thymectomy, I think that's important.
This is a possibility also, yeah.
Thank you.
Okay. You were actually earlier on time, so we have time for another, was there another question, or? No? Is it the same question? I have I think we're running out. I'll ask you later. Thank you very much. You put the red thing.
Yeah. All right, then we welcome Dr. Morroni from Italy to the stage. He's postdoctoral researcher, and he's going to talk about one of my favorite cells, the B cell.
So good day, everybody. Thank you, Jacopo Morroni. Thank you for the opportunity to share a part of the results we had and the work we're conducting at Policlinico Gemelli. I will tell you about the identification of a novel metabolically active B cell cluster in myasthenia, in refractory myasthenia gravis. We talk about B cells and MG, so we have some open issues. Despite being a prototypical B cell mediated disease, we have no consistent B cell signature identified among MG patients. Patient response to immune suppressor and even B cell depleting agents, as we know, is highly heterogeneous. And by now, in general, we have no cellular or serum marker to monitor or predict therapy efficacy. And with those novel therapies emerging each year, there's quite a big issue for clinicians. So the aim of our study was to deep phenotype B cells in myasthenia gravis to uncover novel signatures associated with disease status or therapy response. This is how we stratified our patients. They're all positive for anti-acetylcholine receptor antibodies. We have naive patients, patients in clinical stable remission, refractory to conventional immunotherapy, and patient in minimal manifestation or pharmacological remission, and of course healthy control. And very quickly, this is the numbers of patients we used for each of the following analysis. So we began our exploratory phase performing mass cytometry. We employed a B cell focused antibody panel with 33 B cell related markers, and after performing dimensionality reduction and flow cell clustering, we could observe some alterations in MG refractory patients compared to other groups. In particular, we have at least 4 clusters that are differentially abundant in the refractory patients compared to other groups, And our attention focused on cluster 16 for several reasons. First, it's quite an abundant cluster, among the increased in the refractory patient group. And second, you can see it in the red circle here, because it has a quite interesting phenotype, we believe. So it's high in glycosylated CD45RB, that is suggestive of antigen encounter or B-cell recirculation to lymphoid organs, but they're CD27 variable to low, they're CD24 low, they're CD11C negative, so they're not transitional cells, not memory, they're not even atypical B cells. We will call them just antigen-experienced B cells. Even more interestingly, the increase in cluster 16 is accompanied by a decrease in another cluster, that is cluster 20, as you see in the yellow circle, and is a cluster mainly composed of steady-state naive B cells. So the phenomenon we intercept with mass cytometry is some kind of a shift from steady-state naive B cells to those activated antigen-experienced, maybe recirculating B cells. We then performed single-cell RNA sequencing on peripheral B cells, and one cluster immediately caught our attention. I don't know why the colors are so bad, but anyway, you can see it. One cluster caught our attention as peripheral cluster number 4. It caught our attention for several reasons. First of all, because it is accumulated in the refractory patients, and second of all, because it has a very interesting transcriptomic signature with upregulation of metabolic genes, CINA1, LDHA, FABP5, and is characterized by downregulation of CD37, which is quite peculiar, accompanied by increase in MYC, antipoptotic genes, and increase in CD95HC heavy chain as an aminoacidic transporter linked with B-cell survival and plasma cell proliferation. Talking about clusters, I'll be quick here. This cluster shows increased glycolysis, biosynthesis of amino acids, pyramid metabolism, and antigen processing and presentation, meaning those cells are somehow cooking up something. Intriguingly, this cluster persists unchanged after rituximab administration. You can see on the left in blue the B cells from a refractory patient before rituximab administration, and on the right in yellow the same patient, B cells from the very same patient 1 year after rituximab administration. The patient did not respond. And the interesting thing is that the overall B cell signature, transcriptomic signature, is totally changed even 1 year after rituximab administration, except for our cluster that remains unaltered, unchanged, also, as you can see from the bubble graphs on the bottom left. Now, we went on, we performed single-cell RNA sequencing on B cells isolated from the thymus of several patients. We employed one non-MG thymoma, one MG naive with hyperplasia, and one MG refractory patient with thymoma 6 months after rituximab administration, and a patient who did not respond to the rituximab. And what we observed was that very similar cluster, we will call it thymic cluster 1, is accumulated in a refractory patient. Very same pathways are upregulated, and among the top 100 upregulated genes, 39 are exactly the same, and the rest is very similar, linked to the same processes. So we have those cells, we call them CD37 low B cells because it's quite a peculiar hallmark, and we ask ourselves 3 questions: who are them, where are them in the thymus, of course, and what do they do? So answer to the first question, we perform a flow cytometry phenotyping, and we observe interesting thing. For example, they are high in glycosylated CD45RB as the cluster we identified in mass cytometry. They're high in CD95 heavy chain as the cluster we identified in a single cell, and we confirmed that they're accumulated in the refractory patients. This is preliminary, but intriguing. They seem to be among the first populations to repopulate the periphery after rituximab treatment. Where are them in the thymus? Well, they answer the spatial transcriptomic. We took thymic tissue from the very same patient we analyzed in single cells, and we built from single cell RNA data a probe set to go at the single cell level spatial transcriptomic. Here I show you the tissue of the refractory patient, non-responder to rituximab after rituximab treatment. We can ignore the thymoma tissue on the right. We will concentrate, we'll focus on the hyperplastic tissue on the left, and we can see that even after rituximab administration, we have structures that are very similar to germinal centers here. If we go in detail, we have 2 main cell clusters comprising those structures. This is cluster 4. Cluster 4 is just as you would expect a germinal center to be. So B cells in the middle, T cells all around, but those B cells are not the ones we're looking for. The B cells we're looking for, CD37 low B cells, metabolically activated B cells, reside at the periphery of germinal center-like structures and near perivascular spaces. How did they get there? What did they do? Well, we don't have to answer for the first question, but we have some clues to answer to the second question. We took those cells, CD37 low B cells and CD37 high B cells, we sorted them and we separately co-cultured them with feeder cells. What we could observe is that after 7 days of culture, CD37 low B cells more efficiently differentiated into antibody-secreting cells, and they produce higher level of EGGs while proliferating less. At last, we wanted to confirm the metabolic activation of this cluster, so we perform a seahorse real-time ATP rate assay on those cells, and we could observe that CD37 low B cells have a higher glycolytic rate at the basal level. So in conclusion, we have those CD37 low B cells that seem to accumulate in MG refractory patients. They seem to recirculate from periphery. They show upregulation of interesting markers as glycosylated CD45RB, CD95, heavy chain, glycolytic transcripts. They seem to reside in the thymus near germinal centers and near perivascular spaces, and in vitro, they can efficiently differentiate into antibody-secreting cells. We have some open questions, of course. One big question, so do those CD37 low B cells directly drive MG pathogenesis, or are they an epiphenomenon of something else, like dysregulated tolerance? And you see some of the experiments we're planning to do in the very next future to try to answer those questions. My time is over, so I thank you all for the attention, and any question or even suggestion is warmly welcome.
All right, thank you very much for this enormous amount of data. So, who has a question? Nobody? I actually have two. So I was wondering, at the beginning of your presentation, you started off saying that you were looking for a biomarker, cell signature of MG. Did you find one now?
Well, we did find this accumulation of those cells in the refractory patients. We're checking now if they're linked to also rituximab response or not, since they seem to repopulate the periphery in non-responders, but this is preliminary still. So we have this association, and we have, truth be told, several other associations with conventional immunotherapy. So we understood that the MG refractory patients, even in comparison to the patients who are under therapy now, but respond to the therapy, have several B-cell markers, several B-cell clusters that are altered. So these clusters, we believe, might be one of the markers.
And did you check if these, especially these CD37 lows, are also antigens specific?
We are testing it right now. We made a preliminary testing, but probably we didn't have enough cells because they're quite a rare population. They were expanded in refractory patients, but refractory patients usually have low B-cell levels because of the immunosuppressants with
you don't see you don't see acetylcholine receptor
not
antibodies
yet.
In your cultures?
Not yet, not specific antibody, not yet, but we are repeating this experiment with larger amounts of cells since the frequency in periphery is very low, as we know.
All right. Thanks a lot.
Thank you.
There are no more questions. Then we move to the next.
Okay, thank you very much.
Oh, sorry, Laurent.
Oh, one more? We are
sorry.
Okay.
Go ahead.
No worries. Wow, what a lot of work. Very impressive.
Thank you.
What do you think could be driving this the CD37 low phenotype? Is this maybe driven by the higher metabolic rate, or vice versa, or is it concomitant? What do you think?
I would say that it's probably just associated. I mean, when B cells, usually when B cells shift their metabolic rates, they're going toward activation. And we know that, of course, activated B cells may express lower CD20, for example, and also CD37, since they are in the same tetraspanding complex and their expression is associated. So I think it's something that is just going on in the cell, and we can see it both from downregulation of CD37 and the metabolic alteration.
Thank you.
Okay. You're welcome.
Thank you very much.
You.
So our next speaker is Dr. Xia Huang, who is a neurology resident from Fudan University in China, who will talk about a prospective study on myasthenia crisis.
Good morning, everyone. It's truly an honor today to share the findings of our multi-center prospective study on myasthenia crisis outcome. As we all know, myasthenia gravis is a life-threatening status on MG that requires mechanical ventilations. Highly cited research in this field included the IVIG versus plasma exchange trial, German multi-center studies, and the MC prediction models. Despite these contributions, mostly focused on acute management and in-hospital outcomes using retrospective designs, a question remained unanswered. What happens to myasthenia gravis patients after hospital discharge in the long term, including all-cause mortality and chronic complications? This is the knowledge gap our prospective study was designed to address. From 2018 to 2024, we screened MC cases across 11 university hospitals. After applying rigorous inclusion criteria, we enrolled 277 patients. First, we analyzed in-hospital outcomes, all-cause mortalities, and chronic complications. Second, we report the MGFA post-intervention status at 1 year after Myasthenia gravis, and identified predictor factors through multivariate regressions in 178 episodes with complete 1-year follow-up. Notably, we defined the variable outcome as AChR, PR, MM, and improved status, which means greater 3 points QMG improvement. And we define the unfavorable outcome as unchanged worse and exacerbations at diet of MG status. Our Chinese MG cohort showed distinct characteristics with an average age significantly younger than German data, that is 52 versus 67 years old. Antibody testing revealed 18.3 were AChR positive, and nearly half were thymoma-associated MG, that is higher than that in Western populations. We also recorded disease duration, MGFA classifications, triggers, and the Charlson Compatibility Index, which is a scoring system for conditions such as heart failure and cancers, diabetes, etc. at baseline. For in-hospital outcome, MC patients require 15 days of mechanical ventilators, 20 days of ICU care, and 35 days of total hospitalizes on average. Notably, mask-positive patients demonstrate significantly shorter ICU stays compared to AChR-positive patients. At discharge, we can see only 32 of patients could return to independent living, highlighting the extent recovery period after MC. This prospective cohort showed 5% in-hospital mortality, lower than most retrospective studies, including our 11% historical data. Annual rates decreased from 8 to 3%, with a peak at 9.5% during the COVID-19 pandemic in 2023. How about the all-cause mortality? The number is 15%. The primary causes of death included septic shock, multi-organ failure, and so on. We found important differences among subgroups. Patients with AChR-positive Myasthenia gravis had significantly worse survival outcomes compared to other antibodies subtypes. Among it, TMG and late onset MG patients exhibited a progressive decline in survival rates over time. This is different from the early stage studies suggest that masked MC patients had the worst prognosis. But in our cohort, it was actually the AChR-positive patients who faced greater challenges. Why? Because in our centers, each masked MC patient received immediate plasma exchange combined with rituximab, and remarkably, nearly every one responded well to this approach. Let's move into the chronic complications. The most concerning was infections occurring in 15% of patients, affecting multiple systems. Other notable complications including GI ulcers and osteoporosis in 5% of patients. Then let's see the 1-year outcome after MC. Approximately 18% of patients achieved what we classify as favorable outcome, and the proportion of reached minimal symptom expression status was 53 points. Then we identified 4 independent predictors of unfavorable outcomes by multivariable regressions. Age, thymoma presence, Charles Combability Index greater than 3 points, high ADL scores of ventilations. To summary, this study provides the first prospective follow-up data on myasthenia gravis outcomes. And Chinese MC cohort exhibits younger age and a higher prevalence of thymoma-associated myasthenia gravis. In-hospital mortality is reduced from 12% to 5%. And our survival analysis showed AChR MC tend to have worse outcome. And of course, managing post-MC complications, especially prevent infections, is very important. And as just mentioned, we identified 4 independent predictors of poor post-crisis outcomes in MC patients. And this study highlighting the importance of disease monitoring and timely intervention in improving the life quality of MC. Finally, I gratefully acknowledge the multiple-center MC team led by Professor Zhao and Professor Ruo. The multidisciplinary team, including the surgery and the doctors for plasma exchange. And we also thank patients and their families. Thank you for your attention.
All right. Thank you very much. Are there any questions? I see Ali going to the mic. Oh, and here, number 2 first. Go ahead.
Okay.
Yes.
So, nice presentation. So, what exactly is that you define as myasthenic crisis? So, did your patients have score, or so? How was myasthenic crisis defined?
Of course, have trouble respiratory breathing and lead to the help with mechanical invitations, and not only QMG scores, because the breathing muscle is the most dominant weakness. And we also perform the blood test of the
oxygen?
Yeah, oxygen and the percentage of CO2.
Oh, CO2.
Yeah.
Okay, thank you.
And Ali?
Hi, I have a couple of questions. So one is, your total duration of hospitalization was quite extended, so that's great, you had a long observation period. What was your readmission rate for patients once they did get discharged from the hospital? And the second question is, do all patients return to their baseline function after experiencing a myasthenic crisis in your cohort?
I answer the second question first. Not every patient return to their baseline, of course, and only 15% reach minimal expiration status. And the first question, could you please?
Your readmission rate from after discharge.
How many patients came back to hospital after the crisis?
Almost 18% come back to our center because it is a prospective studies, and we try our best to include all the patients and give them follow-ups.
I think the question is, did they have to come back to hospital to be admitted to hospital for another crisis or another exacerbation? Were they very sick again, or they were stable?
Were sick again. And we also perform a regular assessment such as QMG and ADL scores. Even he's stable, not need to the acute management.
Okay. Thank you very much. We don't have time for more questions. We'll move to the next one.
All right. So there's a slight change in program now. Dr. Corral was unable to join us, unfortunately, but she found a very worthy replacement in Ines Muntadir. She's a PhD student at Istanbul University, and she'll talk about IL-4 receptor targeting in a MuSK-MG mouse model.
Thank you. Yes, thank you. So good morning, everyone. So as she said, Gizem Corral was supposed to do this presentation, but unfortunately she couldn't come. So I don't know if I will be able to answer to all your questions, but I will do my best. So we were interested in studying the effect of silencing the IL-4 receptor alpha via shRNA lentiviral particles treatment in experimental autoimmune Myasthenia gravis. So why we were interested in this pathway, IL-4 receptor? First, because IL-4 is involved in producing IgG4 antibodies, and it's involved in the class switching of these antibodies. And we know that IgG4 are involved in the development of Myasthenia gravis. Especially in chronic antigen exposure, such as in Myasthenia gravis. So that's why it would be interesting to target this IL-4 receptor, more particularly the subunit alpha, to decrease this production of pathogenic IgG4 antibodies. And that's why we were interested in the shRNA therapy to target this receptor. But the thing is that mice don't produce IgG4. So we looked instead at the IgG1 because we know that they are the analog of IgG4. They are functionally similar. As we can see in this study, in Myasthenia gravis mice, there is an increased level of IL-4 and IgG1. So in this study, we decided to look at the IgG1. And previously in our lab, we did a study to target FcRLB in Myasthenia gravis with the same method of shRNA silencing. And we saw that it reduced the clinical severity, and also it decreases the amount of IgG4 antibodies. So it really shows that this method is effective and it's feasible. So that's why we decided to continue with this method, but this time by targeting IL-4 receptor alpha. So our objective was to assess the immunological and pathological consequences of IL-4 receptor alpha knockdown via shRNA technology in a model of experimental autoimmune Myasthenia gravis, EAMG. So for this, we immunized 36 female black 6 mice with ecto-musk protein via active immunization. So we first immunized them and then with the antigen of MuSK in CFA, and then 4 weeks later we did a second immunization, and just after that we injected the shRNA or not, depending on the group. So the mice were divided into 4 groups, including the controls with scrambled shRNA and also CFA as a negative control. I have to say that 2 mice died before sacrification in the scrambled shRNA group. So then we were looking at the clinical score, ranged from 0 to 4, with 4 indicated that the mouse was found dead. And we were looking at the weight, the grip strength, the behavior of the mice. And I have to say that we were 2 blinded researchers. So basically, I was looking at all these parameters and recording my voice, but I didn't know which cage corresponded to which group. And then my colleague was collecting the data in a file. So these are the first results, and we can see in the treated group that the clinical score is significantly better than the control groups, the non-treated and the scrambled shRNA group. So it improves the clinical disease severity. We also looked at the weight and the grip strength, and the results are here again significantly different in orange for the control group. So it was significantly higher than the controls groups. And then we wanted to look at some cytokine levels via ELISA. We didn't see any significant difference regarding IL-4 level, but also interferon gamma, IL-2, or IL-10. And the IgG subtypes against MuSK. So here we clearly see that there is a decreasing of IgG1 level in the treated group. It's not significant, but still it tends to decrease. And it's not the case for the IgG2b and IgG3, where the level of the antibodies is quite similar to the control groups. Then we did some immunofluorescence stainings to look at the neuromuscular junction and the deposit of the antibodies. So here we can see that in the treated group, the neuromuscular junction, the membrane still quite intact compared to the control groups, where it starts to be degraded. And also for the deposit of the antibodies, we have almost no deposit of IgG compared to the control groups. And it was the same for IgG1 with an intact membrane. And we also looked at IgG2b and IgG3, where we have the same observations with a less amount of deposit of the antibodies. We got an unexpected result for the scrambled shRNA, but we are still making some hypotheses we are still trying to understand these results. Okay, so the next steps will be to do some to use other methods to validate our effects, our results with PCR, Western blot, flow cytometry, in vitro stimulation. So it's very preliminary data. But a lot of papers show that it's really interesting to target this pathway of IL-4 receptor alpha, like with dupilumab. Sorry, it's an anti-IL-4 receptor alpha antibody, and they showed in some studies that it allows to decrease IgG4 production, and there are also new experimental antibodies to target IL-4, IL-13 axis. So there are some limitations in our study. So as I said, there's no IgG4 in mice. Even if IgG1 is functionally similar, we have, for example, the Fab arm exchange present in IgG4 is not present for IgG1, and there are also disease model limitations because we cannot observe IgG subclass specific functions, so that's why we would need humanized mice. And yeah, we still have to quantify our knockdown with PCR and etc. So to conclude, we showed that this shRNA silencing significantly improved clinical outcomes in MuSK EAMG by reducing IgG1 levels and deposits. So targeting IL-4 receptor alpha is a promising strategy, and it could be a potential therapy for MuSK Myasthenia gravis. So with that, I would like to thank you for your attention and to thank all the lab members and our partners for making this project possible. Thank you.
Thanks a lot for this nice overview. Are there any questions? Nobody rushing to the mic. In that case, I was wondering, because dupilumab is a IL-4 receptor inhibitor in clinical trials, how does the dosing in these animals relate to what is being used in the patients?
Compared to the patients, I don't know, but we were using 45 microgram per ml for the immunization. Do you know how is it for human? No.
I have no idea, to be honest.
I don't know. I cannot tell you, sorry.
All right. I think in view of time, we'll move on.
Yes, thank you very much.
Thanks a lot.
Okay. Next speaker, Dr. Hae Yoon, who's had an early career neurologist at Yonsei University College in South Korea. And she will talk of risk of death. We see a theme here in Korean patients with myasthenia gravis.
Okay, thank you for that introduction, and thank you for having me here. Today I'm going to talk a little bit about the myasthenic patients in Korea. We did a nationwide population-based cohort study. Okay, so the incidence of MG is known to be around 0.03 to 2.8 per 100,000 individuals. And the global prevalence is known to be around a little over 12 per 100,000, with about 700,000 MG patients worldwide. The mortality in these patients is particularly high in older men, and the leading causes of death were malignancy, cardiac disease, and MG. This incidence and prevalence of MG has increased recently, particularly among older individuals, which is possibly because of higher disease recognition, an aging population, longer patient survival due to various new treatments. And these advances in diagnosis and treatment have led to a decrease in the MG-related mortality, but the presence of various comorbidities with MG and these complications due to treatments make it a little difficult to interpret a mortality when it comes to MG patients. So our study was to investigate the risk of mortality in patients with MG using a Korean national claim database. We used a public database provided by our NHIS, where all patients and all medical institutions are required to be registered. We looked at cases diagnosed with the diagnosis code for MG between 2005 and 2020. We used a age and sex matched control group without the diagnosis code for MG at a 1:5 ratio. All-cause mortality and demographic, clinical, and treatment variables were included in our database. This is a pretty big table, but to summarize, the characteristics of like 21,000 patients with MG and 108,000 control group were compared here. The patients with MG had a higher prevalence of comorbidities, including diabetes, hypertension, hyperlipidemia, osteoporosis, chronic kidney disease, COPD, myocardial infarction, atrial fibrillation, heart failure, stroke, and cancer. We then compared the cumulative incidence of all-cause mortality. The graph here shows that the MG group has a higher mortality rate compared to the control group. The hazard ratio was 1.63 after adjustment for age and sex. After adjustment for the comorbidities, the hazard ratio remained 1.41. So even after adjusting for these variables, the MG group had a around 40% higher mortality rate compared to the control group. The demographic and treatment-related characteristics of the 2,000 deceased MG patients and the 19,000 surviving patients were compared as well. Patients who died had a significantly higher mean age at diagnosis, and there was a higher proportion of men, and the CCI score, which was mentioned earlier, a score that signifies like various comorbidities. These were higher in the deceased group. The deceased group also had a higher prevalence of multiple comorbidities. MG crisis was also higher in the deceased group. Regarding MG treatment, the mean daily prednisolone dose was higher in the deceased patients compared to the survivors, and the use of non-steroidal immunosuppressants, intravenous immunoglobulin, plasma exchange, these were all higher in the diseased group as well. On the other hand, thymectomy was shown to be protective against mortality here. We did a multivariable analysis, and here we see that age and male sex, as well as various comorbidities, was associated with increased mortality in the MG group. And for the factors associated with the MG disease itself, thymoma and high-dose prednisolone was associated with a higher risk of death in MG patients. Myasthenic crisis was a significant factor. Thymectomy was, again, protective. And here we show a graph showing that myasthenic crisis was a significant factor in mortality. Earlier, the hazard ratio was over 3, so there was more than a 3-fold risk of death in the patients with myasthenic crisis compared to those MG patients who did not have myasthenic crisis. Death due to MG itself was observed in around 1.18% of MG patients, and the most common cause of death in both MG and control groups was cancer other than thymoma. But even malignancy other than thymoma was significantly higher in the myasthenia group compared to the control group, which was pretty interesting for us. Other causes with significantly higher frequencies in the MG group included respiratory disease, thymic malignancy, infectious disease, and injury. A lot of these could be explained by MG treatment or MG disease itself. The graph on the left shows what I just mentioned earlier, malignancy other than thymoma, myasthenia gravis. These were all major causes of death in patients with MG. Because myasthenic crisis was a very significant factor in the death of MG patients, we looked at the causes of death in the patients with a history of myasthenic crisis. Here, there was a very high frequency of myasthenia gravis itself as the cause of death. This group also showed a high frequency of deaths due to thymic malignancy and malignancy other than thymoma. Okay, so to summarize, this study showed that the mortality rate in patients with MG in Korea was higher than the general population. Various characteristics, comorbidities, MG-specific factors were identified as independent risk factors for death. Myasthenic crisis, thymoma, thymectomy, these were closely related with mortality. And we think that these findings show the need for further research for early identification of high-risk patients to reduce mortality and improve long-term outcome for these patients. Thank you very much.
Thank you. I see people standing up who came to the mic first. Jan and then Stephen.
Thank you very much. Very interesting study with very large numbers and many associations to give food for thought. So I wonder, so what is the order of the events? Is it that myasthenia patients, that we miss something, they are at risk for some diseases, and is that correlated to the susceptibility of the myasthenia? Or is it all consequences of the treatment that we give these patients?
That's pretty much mixed, I think. It's very, this was just a cohort study. We don't know the course of events. Maybe looking into the more specific dates of like cancer diagnosis and stuff, I think that is things for further research in the future.
Thank you very much.
Stephen?
Over here. Thank you very much, very nice. If you correct the thymoma data for death by all malignancy, because thymoma is associated with more malignancy as well as itself being potentially fatal, does it still have an effect on mortality?
I don't think we've tried that. We just, so far, we've only looked at the effects that thymoma and malignancy has on MG itself. We don't know for certain if malignancy and thymoma are related to each other. Again, thank you for that suggestion, and we will look into it in the future.
Sorry, we have to.
You can take them for the break.
Yes. All right, thank you very much.
Thank you.
Thank you.
And we'll move on to our next speaker, who is Sofia, and I'm checking, Natsuko Gotsamani. Yes? Good. And she's going to talk about MuSK antibodies of other subclasses and isotypes. The floor is yours.
So, good morning, everyone. Before diving in, I would like to thank MGFA for the privilege of sharing our work today. And up until yesterday, I was very excited to share IgA anti-MuSK as a new finding, but Dr. Massi stole my thunder. But nevertheless, I will show you what we have found in my lab. No? Okay, sorry. So you already know about how MuSK contribute to AChR clustering, so we'll skip this. And you also know that in MuSK-MG, IgG4 is the predominant antibody that targets MuSK and doesn't interrupts the whole signaling cascade and provokes muscle weakness. But also IgG1, 2, and 3 anti-MuSK are found in a subset of MuSK-MG patients. These non-IgG4 antibodies were reported over 2 decades ago, but in their pathogenicity and clinical relevance was reported it was actually investigated in the last years. And I'm sorry. And actually, the recent study suggests that these antibodies are promising biomarkers for the clinical severity and for the clinical severity. So, in our lab, we use RIPA in order to screen suspected MG patients, and so far we have found 234 samples from 142 patients positive for anti-MuSK IgG. But here is the catch. RIPA shows only that IgG exists, but it doesn't show what subclasses are at play. So we were wondering, are our MuSK positive samples have also, is only IgG4 driven, or IgG4 coexists with the other subclasses? But we didn't stop there. Recent findings, recent publications showed that IgA autoantibodies are present in neuroimmunological diseases. So we were wondering, does IgA also target MuSK in MG? Our live CBA was positioned to test this. Let's visualize what we have found. 60% of our samples were exclusively IgG4 driven, and 38% were a combination of IgG4 and the other subtypes, as you can see in the bar chart. But I would like to focus on the other subsets that we found, because we also identified IgA anti-MuSK. 70% showed it was positive for IgA, IgG4, and the other, in combination with the other subclasses of IgG. 7% was IgA and IgG4 double seropositive, but the most surprising outcome was that we found 4% only IgA anti-MuSK positive. The remaining 18% was negative, probably due to the limited sensitivity of CBAs in low-targeted antibodies, but we also found some of these that were IgG-Fc positive. Coming back to IgA anti-MuSK, I would like to show you how it looked like under the microscope. So, using serum 1, screening it, serum 1 is one of our anti-MuSK IgA only positive samples. You see clearly that it has anti-IgA signal and a similar pattern as well for anti-IgA1 subclass. It was negative for anti-IgG4 and IgG-Fc as well, ruling out cross-reactivity. Critical controls confirmed that our signal was real. So healthy control was negative, and there was no reaction to aquaporin 4 cells using the serum 1, meaning transfection success. So our next question was whether IgA is targeting the same domain in MuSK as IgG4 anti-MuSK, so we did a dual labeling with a serum for positive for both IgG4 and IgA. And you see here with red IgG4 localized on the MuSK-rich membrane. And a similar pattern you see with IgA in green. And merging these two, we clearly see, especially in the zoomed in set in the bottom row, that there is nearly perfect overlap. So, could IgA contribute to MuSK MG pathology? Let's recap some key facts. So, IgA is because this can be due in there is IgA1 and IgA2, and it can be in bloodstream or in mucosal secretion, and it can either as a secretory antibody play a role in immune exclusion, or as a sera antibody interact with complement response or with IgA-Fc receptors of myeloid cells. IgA also plays a role in autoimmune diseases such as IgA nephropathy and other, including the skin vessels or the joints. And as I previously mentioned, it has been found that IgA also can target, for example, MOG in a subgroup of patients with CNS demyelination, suggesting a pathogenic role. So coming back to our samples, which was only IgA positive, here we have 11 samples. In purple you see 3 samples from the same patient, and all of them were their IgG anti-MuSK titer was very low, close to the positivity threshold of 0.03 nanomolarity. As I mentioned, they were not positive for any IgG subclass, and screening for IgA1 and IgA2, we found that only IgA1, they had all IgA1 subclass. And we wanted also to see if they have antibodies against AChR or LRP4, and we found 2 samples positive for AChR, and both of them we identified later on that they were double seropositive MG patients. In order to assess and validate our findings, we also included 157 controls, samples from different kind of control groups. We have 3 non-MuSK MG, 8 other neurological diseases and healthy control group. All of them were negative for IgG4, IgG3, and 2, but you see that some of them were IgA positive. However, if the AChR positive samples may reflect double seropositive MG. And while MOGAT, the 2 MOGAT samples, they had also IgA anti-MOG antibodies. And regarding the 4 healthy samples, they were also reacting to additional antigens, and maybe all these last 2 things may reflect non-specific background. Another interesting fact is that it came from the tracking of IgA and IgG levels in patients from our IgG-only positive patient cohort who had multiple samples. And you can see here, for example, that in the beginning we have IgG samples, IgG subclass is positive for all, and it goes lower and lower, and at some point they disappear, and then we have suddenly an IgA positive sample, and this disappears as well. Here, the difference is that IgG4 and IgA coexist, then the IgG titer goes low, and IgA remains, it disappears again, and the IgG titer rises again and lowers again. And these 2 patients have a similar pattern with the above, but still we have a limited sample size, so in order to conclude anything or identify any patterns, so we need to investigate more on that. And as a future work, of course, we want to see quantify the IgA titer of anti-MuSK, to collect the clinical data of these patients and correlate it with antibody dynamics. And of course, to do some functional experiments to understand better the IgA pathology in MuSK Myasthenia Gravis. And with that, I would like to thank you all for the attention, and I'm happy to take any question.
Right, are there any questions?
Hi, nice work. I was wondering about the patients with the AGA for mask only. Have you tried to pre-adsorb with the mask protein or lysate of cells expressing masks to see if the binding persists or not?
To be honest, we didn't try it in general, but we tried actually, we are actually on work to only exclude the IgG4 antibodies from the IgG4 and IgA positive samples. So in order to see if we still have like a positive reaction for IgA antibodies in mass. But in general, just to subtract it and see if it's still positive, we didn't try it yet, but it's a nice experiment. Thank you.
Okay.
All right. One very short question.
Yeah. Could you use this information? I know you're a laboratory, so you don't have all the clinical information, but if you could go back to the clinicians and to see how these patients are being treated. So assumption would be that the ones with the IgG4 antibody would well react to rituximab, but the other might not, or to a lesser extent. Do you have any clinical information from the referring doctors?
Yes, so also Dr. Maestri explained that yesterday, so most probably they also really appear after the rituximab treatment. We are still collecting clinical data, but what we see is actually all these patients were treated with rituximab. We don't know yet exactly the time point, so that would be an interesting thing to see if it's somehow correlated with this treatment. But I cannot give you more information about it because we don't have it yet.
Right.
Thank you.
Thanks a lot.
Thank you.
Okay. Moving on, I'd like to invite our next speaker, Dr. Paola Calvacante from Italy, who's going to talk to us about some of her research in biomarkers for unresponsiveness to immunosuppressive drugs.
Good morning, everybody. First of all, I would like to thank the steering committee for giving me the opportunity to present my data at this great conference. As we know, myasthenia gravis treatment has been evolving significantly in recent years thanks to the advance of targeted therapies. It's now clear that the conventional one-size-fits-all approach based on selective chronic immunosuppression does not allow to achieve complete stable remission in most patients, and a proportion of them are refractory to immunosuppressive drugs or become refractory over time. So inter-individual variability in the clinical response to treatments highlights the need for a shift from current medicine to personalized medicine based on biomarkers or combinations of biomarkers able to predict the most effective treatment in each patient, including biological drugs. This shift represents a big challenge, but with strong potential to improve therapeutic success and patients' quality of life. As part of this process towards data-driven, personalized, targeted therapies, we focused on a key step consisting in identification of biomarkers associated with high responsiveness to immunosuppressive drugs that could be suitable for selecting patients who could benefit from early treatment with biological drugs. Non-coding RNAs are ideal candidates as personalized medicine biomarkers for immune-mediated disease, such as myasthenia, because they are able to modulate the expression of genes implicated in both immune response and drug metabolism. Changes in their levels can reflect distinct pathophysiological states, allowing monitoring of therapeutic efficacy in each patient individually over time. We know that macroRNAs has been widely implicated in MG pathogenesis, and some of of them have been described as biomarkers associated with distinct disease subgroups. So our aim was to identify non-coding RNAs, including both macroRNAs and long non-coding RNAs which interact each other, associated with a responsiveness to conventional immunosuppressive drugs in MG patients. We performed the whole marnome and transcriptome analysis by macroarrays in peripheral blood cells of AChR positive MG patients, whole female and with early disease onset, to have an homogeneous group of patients treated with immunosuppressive drugs and stratified as a responder and non-responder according to specific clinical criteria based on clinical score improvements. And we included in the analysis a group of healthy controls. After integrating marnome and transcriptome data, we were able to identify dysregulated RNA networks and selected the key molecules to be validated in a wider cohort of AChR positive patients by qPCR. In the discovery phase, we identified the 10 key macroRNAs and 281 transcripts, including both gene transcripts and long non-coding RNAs, differentially expressed between non-responder and responder patients. Of note, responder patients were more similar to healthy control than the non-responders in terms of expression profiles as a result, luckily as a result of the normalizing effect of the treatment. We developed a computational pipeline to integrate all the data and reveal the networks of RNAs dysregulated in non-responders versus responder patients, and we found by gene ontology analysis an enrichment in signaling pathway involved in B and T cell proliferation and activation, particularly TGF-beta signaling, PD-1, and tyrosine kinases signaling, and pathways implicated in cell proliferation. In the validation phase, we found a significant downregulation of MIR150 and 423 in non-responder compared to responder patients. The levels of these macroRNA were able to discriminate between the two groups of patients and negatively correlated with QMG, suggesting that lower levels of these miRNAs in PBNC associated with high responsiveness to immunosuppressive drugs and increased disease severity. Of note, MIR150 is a key immune-related miRNA, already discovered as a biomarkers for AChR MG. It is a regulator of T and B cell proliferation, and its main target is MIB, a transcription factor which is critically implicated in B cell proliferation and survival, and we found that MIB transcriptional levels were significantly upregulated in the non-responder patients, and these levels discriminated between the two groups of patients, and we suggested that lower levels of MIR150 is able to induce increased B cell proliferation through MIB, and indeed in non-responder patients we found an increased percentage of memory B cells compared to the responders. As regard to MIR423, it is critically implicated in cell proliferation as well by targeting EIF2 and MALAT1, two molecules critically implicated in cell proliferation, and we found that both EIF2 and MALAT1 transcript levels were significantly upregulated in non-responder patients. The levels of these two molecules positively correlated each other, and those of EF2 negatively correlated with the miRNA level, but positively correlated with the transcriptional level of Ki67, which is a proliferation marker. So we suggested a putative link among this regulation of this molecular axis and impaired negative regulation of cell proliferation and resistance to immunosuppressive therapy. So we can conclude that the effective control of T and B cell proliferation due to this regulation of specific RNAs, non-coding RNAs could underlie unresponsiveness to immunosuppressive drugs, and the RNase molecules we identify can be suitable biomarkers for investigation as predictors of unresponsiveness to immunosuppressive drugs, and then the selection of patients that could be early directed towards targeted therapies in the frame of personalized medicine strategies for MG care. I would like to thank all the collaborators and the colleagues at the Neurological Institute Carlo Besta in Milan, with special thanks to the researchers of my lab, Alessia Berni, Maria Cristina Tarasco, Nicola Iacomino, and the neurologists, particularly Rita Frangiamore, Carlo Antozzi, and Renato Mantegazza. And thank you all for your attention.
Thank you very much. Okay. Any questions? No questions? So, as a clinician, how do you see the future of this actually, or when do you see the future for us getting to use this in clinical practice? What's missing there?
So we think that personalized medicine is crucial to be achieved, and particularly it is crucial to move biomarkers into the clinical practice, because they can be helpful to assist the clinical decision making. Our idea is it could be important to integrate both biological and clinical data. So we are working on different sets of biomarkers, not only macroRNAs and non-coding RNAs, but also genetic biomarkers and protein biomarkers to combine all the data with clinical data in artificial intelligence models. We think that the future is in the use of these tools, artificial intelligence modeling tools that can help in the clinical decision-making process, not only as to predict the best treatment option for each patient, but also to monitor disease course under treatment to optimize the therapeutic process over time in each individual patient.
Okay. We're certainly looking forward to that. Thank you very much. And our last round of applause for Dr. Cavalcante.
Thank you.
And our last speaker, and also switching gears, so Dr. Birbaum, Simone Birbaum from France, she's a physiotherapist, and she will talk to us about exercise in myasthenia gravis.
So good morning, everyone. Thank you to the steering committee for providing me the opportunity to present our data, our work, and also I wanted to thank MGFA for coming to Europe, because we're really happy about that, and that gives us more of an opportunity to participate in these amazing conferences. This is patient perspective work, and I really want to acknowledge AChR, who is here, who worked directly with me. She's at the back hiding on this work. So I'm going to talk to you about the facilitators and barriers to exercise in autoimmune myasthenia today, and it's based on a survey study that we did a while back. So just steering gears yet to exercise. So we don't have specific recommendations yet today for exercise in MG, but based on the literature, there is literature out there. We know that exercise is safe in periods of stability, and it does provide multiple benefits, and that's been shown in studies in myasthenia. Improve lower limb strength, walking capacity, improved daily function. But also, there is so much exercise literature out there in the general population and in other chronic diseases. And obviously, patients with myasthenia can also reap those benefits. And that's really important to consider, because we can't always reinvent the wheel. So I just want to highlight those. Decreased mortality, decreased risk of multiple chronic diseases simultaneously, improved well-being, reduced anxiety and depression, which we know is really important in myasthenia, pain, improved societal participation and insertion. Exercise, in my opinion, is the magic pill, but only if you do exercise. So we know also from several observational studies in myasthenia that patients with myasthenia do large amounts, are very sedentary, They also, when they are active, the activity is at a lower intensity, and when they do increase that intensity, they fraction their time in those higher intensities. But looking at those activity levels, we didn't find an association with disease severity. So we can't, based on these studies, we can't really explain why they're doing less and lower intensity, and why generally they're less active than other people. We also know that, and you probably know this as well, there are a lot of questions surrounding exercise. There's a lot of fear and apprehension from the part of patients, some clinicians as well. Is it safe? I'm scared. Which exercise can I do, and how much can I do? So we wanted to understand. This is a collaboration, as I said, with the Patient Association from the AFM Telethon. And our aim in this study, the MyAX study, was to identify factors that could influence exercise participation for adults living with MG in France. So it was open to all of the myasthenic syndromes, because the Patient Association represents all of the myasthenic syndromes. But today I'm just going to present a tiny little piece of the puzzle on the autoimmune cohort. So it was a cross-sectional study, observational, via an online survey on a secure platform. The platform was open for 6 months, and we collected a lot of data on socio-demographic information, health and MG info, exercise-related information, and also several validated PROs. We were lucky to have a participation from all regions in France. We had 455 responses, female predominance, mean age of 57, but it's important to note that there was a large range of ages from 19 all the way up to 90. There wasn't at all a focus on the younger age groups being online. The mean disease duration was 16. And based on the response from participants, whether they participate in exercise or not, we divided the group into two. So exercise was really was defined as it's a subset of physical activity, so it's not the incidental exercise that you do in transport or washing the dishes, the domestic chores. It was actually exercise that's planned, structured, active and obviously and voluntary, with the aim of improving or maintaining your health or fitness. So we had more responses from the exercises and less from non-exercises. And I just want to highlight, I've only provided some information about the cohort. 80% had generalized MG, and in those that had ocular MG or no current symptoms, there were more in the exercise cohort than the non-exercise cohort, which can explain some of the following data. And I just want to also highlight that it is a cross-sectional study, so I'm not at all claiming any cause or effect here. But exercises did report stable disease. They were more satisfied with their current symptoms. They had a lower mean MG-ADL score, a higher quality of life, and lower fatigue rates. When we looked at the reported symptoms, both groups were similar. So you have in green the exercises, in blue the non-exercises, except that the non-exercises had more frequently reported lower limb symptoms and respiratory symptoms. When we asked them how they feel about their symptoms with respect to exercise, they both agreed that the three major symptoms which bothered them during exercise, or potentially would bother them when they exercise, the lower limbs, upper limbs, and breathing. When we asked the exercisers about their exercise habits and how MG affected that, there were some that began exercising after their diagnosis, a very small percentage. There were 11% that didn't change their exercise routine, and there were more than half that exercised less since being diagnosed. With respect to the non-exercisers, 2/3 stopped exercising post-diagnosis. 77% claimed that they want to exercise today, but half of them are scared to exercise. So when we looked at the key barriers, for both groups, health and symptoms was the main barrier, was the most frequently reported for both of them, more in the non-exercises than the exercise, unsurprisingly. Looking at some other barriers that were reported, they were allowed to write down as many barriers as they liked. There was no limit. Lack of specialist supervision was the second most frequently reported barrier for the non-exercisers. As well, in third place was the fear of consequences on their disease or their symptoms, a lack of exercise knowledge, lack of motivation. For exercisers, weather conditions was a clear barrier for them, for 40%. And then there were intrinsic factors also, such as lack of time and also lack of company of others to exercise. When we looked at the facilitators, the main factor that could facilitate exercise in the non-exercisers was having specialized supervision, so kind of the lack of supervision and having supervision, so that makes sense. An improvement of health and symptoms was stated by both groups. Having knowledge about exercise or being more informed about exercise was a really important factor for non-exercisers. Having company was important for 30% of both groups. Some practical aspects were important for non-exercisers, and support from family and health professionals is an important enabler for exercisers, as well as exercising regularly, and some didn't have any particular enabler to report. So in a nutshell, since being diagnosed with MG, a lot of people have reduced or stopped exercising, but they do have a strong desire to exercise, but there is a lot of fear and apprehension. So I've highlighted some of the barriers and facilitators that I think we can modify or we can help patients with. So the lack of special supervision, the fear, and the lack of knowledge, as well as the support from health professionals and families. So I feel like that's something, that's a bit of a takeaway message for now until we've finished analysing everything. Because I think they're really something that we as a community can work on. It's going to take some time, it's going to take some energy, but that's something that I think is something we can work on to help patients get exercising, and which can improve their lives in many ways, physically, their health as well as mental health. So I've just mentioned a few suggestions, just on a positive note, an active note, something to do. Just if you can, I know we talked about how long consultations are already in clinics, but just inquiring about exercise habits, research has shown that makes a real difference. Educating and answer questions where possible. There are some resources out there and some studies. It might not be onto you to do that, and you might not have the time, but feel free to refer on to other appropriate healthcare professionals as much as possible. And of course, anything is better than nothing. Take the pressure off people to have certain goals and meet certain recommendations. Start bit by bit. That's really important. It needs to be tailored, individualized. People have comorbidities, and other different functional status. And I just want to leave on, whilst we're working towards a world without myasthenia gravis, I am working towards, while we're waiting for that to happen, I would like for MG and exercise to coexist. And I just want to thank everyone for your attention, and I also really wanted to thank all the people who've worked on this study, the patients for the help in creating the survey, all the co-investigators, and also all the assistants in diffusing the online survey to reach out to as many people all over France as we could. Thank you.
Questions?
One shot.
Okay, we're a little bit running late. Any questions? If no pressing questions, Jan, you have to run, exercise towards the microphone.
So the people who were exercising, were they already exercising for a long time during their whole life before they got myasthenia? So are they made used to that, or because 2/3 stopped? But
yeah, so the exercise, the length of how much, the time they'd been exercising prior to the diagnosis, I don't have that info, yeah. Okay. It was really hard to choose how much, because it was self-report data, and the recall bias, and we do have some data on obviously the time of onset of the myasthenia, so we can look at those types of things, but yeah, we don't have that data, I'm sorry.
Okay, thank you.
Okay. No, so sorry about running, but you can connect with Simone after.
And bon appetit.
The break.
Okay. Good summary.
Yes.
And thanks all the speakers again for this session.
Thank you very much for the great talks. Round of applause.
Yes.